Hello everyone, my name is Jiajun Li, and today I will demonstrate how to perform principal component analysis, which is short for PCA, on a single-cell RNA sequencing dataset. Well, this dataset comes from Cargo, containing 5,697 cells and 1,186 genes from a 57-year-old male donor. Our goal is to explore how PCA helps reduce the dimensionality of such high-dimensional gene expression data while retaining most of the information. We will start by importing necessary libraries: ScanPy, Pandas, NumPy, and MATLAB library. Now we will load the data. We will read the expression matrix and the corresponding gene and barcode information. Well, this matrix contains the gene expression counts with rows as cells and columns as genes. After loading, we assign gene names and cell barcodes to our object. This gives us a structured dataset ready for pre-processing. The uploader already filtered the data for cells that appear to be healthy and alive, so we conduct the analysis without going through the filtering steps. However, we still need to remove the low-quality alive cells that express too few genes, and genes that may appear in too few cells. Then, we calculate mitochondrial gene percentages, since high counts may indicate alive but damaged cells. Here, we filter out cells with more than 5% count reads. Next, we will normalize the total counts per cell to make expression levels comparable, and log transform the data to stabilize variance. Select the top 2,000 highly variable genes, and standardize the data before performing PCA. And now, we will select 50 PCs and check how much they can reserve the raw data information. Well, as you can see, here's the PCA table, and now we will use it to draw its scree plot. Well, this scree plot shows how much total variance is explained as we add more principal components. We can see that the elbow point can be 7. This means the first 7 components explain only about 22.6% of the total variance. Well, this indicates that while the data is very high dimensional, most of the variation is spread across many components. Now we will try 500 principal components. I think this might seem to be too large for PCA, but there's over 1,000 variables to select, so I think it's worthwhile to have a try. And here's the PCA table number 2. Well, and now we will use it to draw a second scree plot. Well, when we look at the cumulative variance curve again, we can find that around 500 components are needed to explain 80% of the total variance. This result is consistent with what we observed in the report, that glioblastoma single-cell data is highly complex and requires many principal components to capture its variability. From this project, I learned the importance of normalization, scaling, and data quality control before running PCA. I also realized that the biological datasets are often much more complex than synthetic ones, requiring careful interpretation. And last part is the advisor's help. I completed this project with advices from Kai Shen, who provided valuable suggestions in writing logic to me, and also advisor. Thank you for watching.